Thank you so much for coming today. So I want to take you back to when you were 7 years old. Imagine it's been raining, you're indoors, and it's just stopped raining. So you look out of the window and you see the most colourful, vibrant rainbow stretching across the sky. So you turn to your mum and you say, "Mum, look at the rainbow." And she gently replies and says, "My darling, I'm sorry, I can't see it." So that is my first memory of my mum losing her sight. Over the years, I helplessly watched her go blind. She can only now see light and dark, and even that small glimmer of light will diminish in the coming years. I couldn't help save my mum's sight, but I have dedicated my life to trying to help others in that position. So hopefully one day I can give them hope of a treatment. My name is Mariya Moosajee. I'm a consultant ophthalmologist specialising in genetic eye disease at Moorfields Eye Hospital. I'm a professor of molecular ophthalmology at University College London and group leader of the Ocular Therapeutics and Genomics group here at the Francis Crick. My lab worked tirelessly to understand the disease mechanisms that cause these devastating sight loss conditions, and it is our goal to develop treatments that will one day hopefully prevent, halt, or slow sight loss. It's my pleasure today to give you the crash course on gene therapy in the context of inherited eye diseases. So before I get into gene therapy, I thought it would be important to give you a refresh on basic genetics. So in every cell of our body, we have genetic material, a genetic code called our DNA. And that DNA condenses to form structures called chromosomes that sit within the nucleus of every cell. Along the stretch of our DNA, we have subunits called genes, and we have over 20,000 genes. And these genes provide the instructions to make proteins, and it's those proteins that help our organs grow, develop, and maintain their function. Our genetic code, also known as the human genome, consists of 3 billion letters, genetic letters. Written out, the human genome would stretch approximately 5,500 miles in length. It would take a typist working 8 hours a day, half a century to type our genetic code. It would fill 1 million pages, 5,000 books stacked 200 feet high, or 200 telephone directories for anyone in the audience who still remembers what telephone directories are. I often say to my patients that in those 3 billion letters of our genetic code, if I asked my secretary to type that out, they would introduce X number of typos. So we all have around 5 million genetic changes or spelling mistakes within our genetic code. I have 5 million, you have 5 million, and you have 5 million. That makes us unique. But of those 5 million changes, 10,000 are potentially disease-causing. And it's those disease-causing changes in our genetic code that lead to genetic diseases or a predisposition to them. So, what are these genetic conditions? By and large, they're rare diseases. And as I mentioned, they're caused by mutations that occur within our genetic code. Now, before I go into genetic conditions, especially those affecting the eye, I just want to give you an overview of the anatomy of the eye, just to orientate you for the rest of this talk. So the eye is divided into the front chamber, also known as the anterior segment, and the posterior chamber. In the front chamber of the eye, we have a clear surface called the cornea that allows light to enter the eye. It passes through the pupil, and surrounding that pupil is the coloured part of your eye, the iris, and that changes the aperture of the pupil. Light then passes through to our lens, which helps us focus light to the back of the eye onto our retina. And the eyeball is covered with a tough white outer coat, the sclera. At the back of the eye, the light hits an area of central vision called the macula. This area is what helps us to see fine detail, colour, recognise faces, read small print. And it contains a light-sensitive cell called cones that help us to see that fine detail and colour. Surrounding the macula, covering the whole back layer of the eye, is the peripheral retina, and that consists of a light-sensitive cell called rods. And rods are what help us to see in dim light and our field of vision. We have a nerve that exits the back of the eye, that's called the optic nerve, that takes signals from the retina and sends it to the brain to help us process what we can see. So there are a number of genetic eye diseases, and there are a number of genes that control how the eye forms, develops, and works. And mutations in any of these genes lead to these genetic eye diseases. And there are over 500 genes that are known to date that are implicated in these conditions. There is a vast spectrum of genetic eye diseases. It can range from the very severe, so a child that is born with no eyes at all. This is called anophthalmia. Sometimes the iris, the coloured part of the eye, is affected and it doesn't grow appropriately. This is called aniridia. Sometimes a child can be born with cataracts. It doesn't just affect older people. Children can be born with this through genetic changes, where the lens in their eye is cloudy. And sometimes it can affect that front layer, the cornea in our eye, and it can cause the cornea to become cloudy in a very patterned effect. These are corneal dystrophies. If we look at the back of the eye, you can see an image of what should a normal retina look like. So you can see the optic nerve, that pale yellow circle. That's the nerve leaving the back of the eye, the red blood vessels tracking across the retina, that area of central vision right at the centre, which is slightly deeper in red, and then the rest of the retina, which is a nice orange colour. Now, if the eye doesn't fuse by week 7 of pregnancy, it can leave a cleft on the underside of the eye. And you can see that here in the image, that huge pale yellowy green area, that's where the retina hasn't fused. And this is called an ocular coloboma. Then you have conditions where the cells, the light-sensitive cells, can die progressively through life. And this is an image of a patient with retinitis pigmentosa. They will suffer from problems with their night vision, then a constriction of their visual fields, and eventually their central vision will diminish. And then this is an image of a very rare genetic condition called choroideremia. And the pale yellow background you can see in the retina is where all the layers of the retina have degenerated, and what you're seeing is the bare white sclera through the back of the eye. So these genetic eye diseases can affect 1 in 1,000 people. Children can be born blind, or they can progressively lose their vision over the years, and it can affect adults at any point, and they can go on to lose vision. These are lifelong conditions that are largely incurable, and for the majority, there are no treatments that can help. We hope that gene therapy will offer promise to these patients, and so we get on to what is gene therapy. So I made an animation a couple of years ago just to explain this concept in an accessible format, so I'd like to play that to you now.
Gene therapy for genetic eye disease. Genetic eye diseases are caused by spelling mistakes in our genetic code, also known as our DNA. They change the instructions and prevent normal healthy protein being made, which leads to disease. Gene therapy packs correct copies of the gene into delivery vehicles called vectors. The vectors are injected into the jelly of the eye, called an intravitreal injection, or under the light-sensitive layer, the retina, called a subretinal injection. The vector then releases the gene into the cell. The cells can then produce healthy protein and can work better for longer.
So, a little bit more detail as to what these vectors or vehicles are that can deliver genes to the back of the eye. So, we commonly use viruses for gene therapy, and the virus that is most commonly used currently as an approved treatment and in clinical trials is the adeno-associated virus, also called the AAV virus or AAV vector. Now, the AAV virus is an icosahedral shape. It has a protein outer coat which has spikes on it. And these spikes are what bind to receptors on cells of the target tissue. And this coat is called the capsid of the virus. Now, within that capsid is the viral DNA, and scientists have removed that and replaced it with a healthy copy of the gene that they want to deliver back to the tissues. When you deliver these viral particles to the target tissue, those spikes will bind to the cell surface receptors and it will be engulfed into the cell. It will then travel to the nucleus of the cell, and at that point it will release the DNA and it will be read or transcribed by the cell's own machinery. So that's the principle of gene therapy. Now, there's only been 6 approved gene therapies so far in the world. The first one was back in 2017. It was called Luxturna, or in the UK it's called Vormatrigine Parvovac, and it was the first gene therapy for an ophthalmological inherited retinal disease that was caused by mutations in the RPE65 gene. The second gene therapy was Zolgensma, and this was approved in 2019 for spinal muscular atrophy caused by mutations in the SMN1 gene. Following that, a couple of years later, Upstaza, which is a gene therapy for amino acid decarboxylase deficiency caused by mutations in a gene called DCC. And that same year, Hemgenix released their gene therapy for factor IX for hemophilia B. The following year, Alividis, which was a gene therapy for Duchenne's muscular dystrophy that replaced the dystrophin gene. And the same year, Roctavian for factor VIII hemophilia A. Now I'm going to focus on Luxturna because it's in the eye and we have the most knowledge about its effectivity in the real world. So it still is the only approved retinal gene therapy so far. It was approved in the UK in 2019 for patients with mutations in RPE65, and it was estimated there were only 80 patients in the UK who would be able to benefit from this. The cost for a single administration to both eyes was £619,000. So how did we get to the point of being able to give a gene therapy to our patients on the NHS? So here's a brief history. It starts way back in 1869 with Dr. Theodor Lieber, who was a German ophthalmologist who described a severe retinal degeneration that caused blindness by age 5 in children. This was called Leber's congenital amaurosis. 100 years later, in 1965, the human AAV adeno-associated virus was discovered. 20 years later, Dr. Nicholas Moosajee and Paul Hermanat introduced foreign DNA using the AAV virus into human and mouse cells. providing proof of concept that gene therapy could be a possibility. In 1997, the RPE65 gene was discovered to cause human Leber's congenital amaurosis. And a few years later, scientists at the University of Pennsylvania restored vision in a dog model who had these mutations in RP65. And this dog is called Lancelot. He visited Capitol Hill after he had the gene therapy, as he had restoration of his vision, to prove that this was something that we should push forwards with. Then in 2008, we had the results of the first human clinical trial. And this was a race between the UK and the US. Researchers at UCL had a gene therapy vector, as did those at the Children's Hospital of Philadelphia. Unfortunately, the UCL researchers' vector didn't work as effectively, so they had to go back to the drawing board, but the Americans were able to push forwards into further clinical trials. And they continued until 2017, where we had the results published in the Lancet of this gene therapy. And in this video, you can see a patient before treatment and then after treatment. And you can see that after a year of this gene therapy, she's navigating through the maze speedily, accurately, whereas before treatment, she's stumbling, she's getting lost, she needs to be redirected. And we saw in the treatment group that 65% of the patients passed this maze test at the lowest light levels, which were impossible for them before treatment. So based on this, the gene therapy was approved in the US. And then, as you know, it came to the UK a couple of years later. Now, I had a patient called Leo who presented to me at 18 months of age. His mum and dad reported that he was having difficulty with his vision from about 4 months of age. He wasn't looking at their faces. He wasn't able to track objects. He couldn't see in dim light, let alone in dark light, and he couldn't discriminate between yellow and red toys. What he did love doing was staring at bright lights, at the mobile phone, at spotlights, at the sun. And even in this video, he's only really working between the very bright areas of sunlight. He gets very disorientated in the shadows. We undertook genetic testing for him in our clinic, and we found that he had the changes in the RP65 gene. So he was one of the youngest patients that went ahead with the gene therapy when it was approved. Now, this is a video of Leo's retina. My colleague, Dr. Robert Henderson from Great Ormond Street, is performing a subretinal injection to deliver that gene therapy. And you can see that bubble there with all the viral particles sitting underneath, transfecting, infecting the cells that we need to treat. After the treatment, we saw an incredible result. I'm going to play you a video of Leo a few months after surgery.
Where's the blue fish?
Blue fish.
Can you give Daddy the blue fish?
Yeah,
That's not blue, is it? What colour's that?
that's a pink.
That's pink. That's right.
It's
That's the blue one. Can
no,
I have?
that's not blue. That's green.
Oh, it's green. Can I have the green one then?
Yeah,
Thank you.
Daddy.
It is green, to be fair. Can I have a cup as well?
Yeah.
Thank you. I'll drink my fish. Thank you very much. A nice cup of fish in the morning to make you feel better.
Now, this really is the stuff that you do see on the headlines of newspapers. So Leo's starting vision was pretty poor. He could see the second line on the chart where the red arrow is. And after the gene therapy, he could read down to middle to the low part of the chart where the yellow arrow is. So an incredible improvement in his vision. But unfortunately, after 6 months, his vision started to get worse again. And now he can only see the top letter of the chart, or even worse. So why did that happen? At the time this paper was published, it's a seminal paper. It described the real world experience of patients having this gene therapy. And this is a consortium from Europe that put together 13 of their cases, and all 13 patients developed what we call a retinal atrophy, a thinning, wearing away of the back of the eye following the gene therapy. What did they show? Well, this is just a lovely image. The diamond in the first image is the site of the injection where they delivered the gene therapy. And as you saw that big bubble over the retina, so it detaches the retina and it spreads across that whole area. By 2 weeks at that site of injection, you start to see these kind of mottling changes. That area grows bigger. And then by 1 year, the area where the injection site was, but also the area of central vision has gone black, suggesting that the cells have all died in that area. So after the publication of this, several other reports were published in the scientific literature, also characterizing this side effect, which had never come out through the clinical trials. So the questions we're now asking ourselves: is this an immune response to the viral vector? Well, quite possibly. Is it a response to surgery? Well, scientists have now gone back to monkeys and they've conducted the surgery, giving some monkeys the gene therapy and some monkeys salt solution. And they only see the chorioretinal atrophy in the monkeys that have the gene therapy. So it's unreally likely to be the surgery. Is it a response to that new gene? The eye has survived without this new gene. Maybe putting it in has caused it to not function correctly anymore, to put it into an imbalance. And maybe that's related to a metabolic response going on in the eye. And maybe it's a combination of all the above. But the key thing is, this is something that is happening to our patients. They need to be aware of this before they opt for gene therapy. We need to make sure that we protect our patients and give them the correct information and know the risks before they go forwards. Now, there have been further studies where we've put more patients together, and we know that around 50% of patients who have this gene therapy can have ocular adverse effects, mainly relating to inflammation. Around 13% we have found now will develop that retinal atrophy. So not as stark as we once thought, but still a significant proportion. There are other limitations to using these viruses. They only hold genes of a certain size. So they can hold genes that are around 4,700 letters in size. But at least in ophthalmology and generally, there are genes that are much larger than that. So how can we treat those patients? Trials are having variable results, and so we need to find alternative approaches. And this is what we're focused on here at the Francis Crick in my lab, a non-viral gene therapy approach. So what we're doing is taking a circular piece of DNA called a plasmid. And these plasmids are composed of only human elements. There are no viral components, so we are expecting a much more reduced immune response. These DNA plasmids do not integrate into our genome, and the benefit of these circular plasmids are that they can hold genes of any size. They can even hold segments of chromosomes that may be missing. They have a special ingredient called a scaffold matrix attachment region, or an SMAR. And this SMAR is found within our normal cells, within our nucleus, and it helps our normal DNA fold so it's ready to be read for transcription, to produce healthy copies of genes. When we place this SMAR in our vectors, it prevents our DNA from being degraded. It allows it to be passed on to daughter cells when the cells are dividing. And it prevents it from being silenced, so it can continuously produce the gene of interest. So we are testing our DNA plasmids on various models in the lab. We use zebrafish here at the Crick, mice and patient cells. And I have a collaborator in the US who works with rabbits. So the zebrafish are a great model because actually their eye has a similar structure to humans. They have an elaborate color visual system. So that again is similar to humans, unlike mice that are mainly nocturnal and just use their rods to see in the dark. Zebrafish see all different colors. And we can manipulate the zebrafish quite easily and watch it develop under the microscope. So we injected our DNA non-viral gene therapy vectors into a single cell of a zebrafish and looked at what happened. The vector was taken up into multiple cells across the whole of the zebrafish, and you can see that from the green signal in the slides. But when we took cross sections of the zebrafish eye to actually look at the retina, we could see lovely green fluorescence in the photoreceptor cells, meaning that the gene therapy was getting to the target cells of interest and expressing the protein. We then have access to an amazing technology where we can take skin cells from a patient with a disease and we can convert their skin cells into stem cells and then grow early retinas in a dish, or layers of the retina. So we then went on to do this and we delivered the gene therapy using a very gentle electric pulse to these cells. And again, we were able to see that green fluorescence, indicating that our plasmids were getting to the nucleus of these cells and producing protein. But both methods that I've mentioned are not really applicable to humans. We can't inject these vectors at a single cell stage in a developing embryo, and we can't really give electric shocks to patients to allow these vectors to enter into the cell. So we need to find a way to get our vectors into target cells. Remember, we don't have viral components, so we can't infect cells. So if you're thinking about oil and water, we know they don't mix. So if we added soap, it would help emulsify the two, mix them together. So we've developed a molecular soap, one that contains a combination of fats and proteins, and it's called a lipid nanoparticle. And when you mix that with our DNA plasmids, it forms these beautiful spherical structures, which you can see in the image. And when you put these spherical structures in contact with the target tissue, it binds to the cells and is engulfed, similarly to how those viruses are. It then transports to the nucleus and it enters the nucleus through those pores. And again, the DNA can be read and produce protein. So we then moved on to mice and we gave the mice a subretinal injection, just like Leo had in the video. And where we inject, we can see lovely green fluorescence, where again, it's producing protein. And we followed our mice for a year and we could see lovely signal in the eye up to a year, which is comparable to viral vectors. So the next stage was to go into a slightly higher animal model, because now we're thinking about, can we translate this to our patients? So we've moved to our rabbits. We've injected it again through subretinal injection, and we're seeing lovely green fluorescence in those light-sensitive photoreceptor cells at the back of the eye. So, non-viral gene therapy is now showing promise. It's our responsibility to prove it's safe and effective before we move to clinical trials. We don't want cases where patients are starting to lose vision after we give them a treatment. But there's still a lot more work to do, making sure it's safe, making sure it expressed for a long time, and make sure there's no untoward complications. So to conclude, gene therapy holds great promise. We don't have to rely on viruses, and in fact, a non-viral approach may be the next generation of gene therapy. But, as I mentioned, we need to find safer ways to replace genes that are widely applicable to all patients. So before I end, I just want to signpost you to a really excellent website that we created in the lab called gene.vision. This is an A to Z of genetic eye diseases. It's fully accessible on all speech software, in all languages. It will tell you all about the condition of interest, the latest research, the background behind some of the genetic therapies that are underway, and all the support groups. There are pages for patients and pages for professionals. I just want to say a big thank you to all the patients who have been so brave and continue to be brave, and for inspiring me and my lab to work as hard as we possibly can so that we can hopefully make a difference to them going forwards in the future. And this is just a patient of mine who has a very rare condition, affects 1 in a million, called Almstrom's disease, who is losing vision every day. And it just highlights the need, how we need to push forwards with this research as safely but as fast as we can. So with that, I'd like to thank my lab, my collaborators, all the people in clinic that I work with, and my funders. And thank you to you all for coming today.